potential, but this potential, which is depolarizing, stimulates the release of neurotransmitter, which in turn binds to the neurotransmitter receptors, which then generate excitatory postsynaptic potentials, which in turn generate action potentials around this first order sensory neuron right here. So nerve impulses are transmitted down the pathway that we'll look at very soon. And here are some examples of first order sensory neurons, and this is described on the next page of the notes, but I'm going to use the picture to explain it. First of all, the first one, salty foods, there's not a receptor for salt or a receptor for sodium ions or other salty ions, but what you have are ion channels, like sodium ion channels, that open up and sodium flows in through these ion channels. And so if you're eating something salty, that salt dissolves in the saliva. Any of these have to, dissolve in the saliva for this to work. So one function of saliva is to dissolve the tastants, so they in turn can then stimulate these receptors, because if your tongue were perfectly dry, this wouldn't work. But when the salt is dissolved in the saliva, it flows through sodium ion channels, which depolarizes, makes it more positive, so that's a receptor potential, generates a receptor potential, which in turn opens calcium channels. You see this, for example, in synaptic end bulbs. They have similar calcium channels that open up. An inflow of calcium then stimulates the synaptic vesicles here to release their neurotransmitters, and those neurotransmitters flow across the synapse, bind to receptors in the the first order sensory neuron here, and that stimulates, the generation of an action potential in that neuron. That's how salty foods stimulate the salt receptors, or salty taste receptors. So you have these different receptors. Sour foods are sour because they're basically acidic, or they are acidic. Basically acidic sounds like two opposite things, but they are acidic. And acids, remember, release hydrogen ions. So if you're eating something sour, there's going to be hydrogen ions in that food, and those hydrogen ions flow through hydrogen ion channels, again depolarizing because it's positively charged, creating a receptor potential. That opens up, again, calcium channels that stimulate the release of neurotransmitter, and again, stimulating that first order sensory neuron. The other receptors for sweet, umami, and bitter flavors are actual receptors, not just channels. So these two are ion channels. These bind to specific chemicals. In the case of sugar, sugars bind to sugar receptors. In the case of umami, and somewhere in there, I think in the notes I've added this, I'm going to have to update the notes, but umami flavors are savory, and so like if you're eating broth or soup or protein like meat, there's a lot of protein in those kinds of foods, and those proteins contain a lot of glutamate, which is an amino acid. And so you have glutamate and some other receptors on those umami receptors. And glutamate, in fact, if you think about monosodium glutamate, it is often added to foods that are savory to make them more flavorful, especially like Chinese foods and things like that. Glutamate is in protein, and it binds to its own receptors. So you have sweet receptors with sweet gustatory receptors that bind to sweet, or sugars, I should say. You have umami receptors that bind to glutamate and a couple of other things that are found in protein. And those receptors activate G proteins. You may recall that olfactory receptors, the odorants also activate G proteins. G proteins in turn activate second messenger systems, which is basically a series of chemicals that then activate other processes that result in depolarization. We're not going to get into all the steps, but basically the second messenger system causes depolarization to happen because of potassium channels. Neurotransmitters are released, and by the way, you might think, well, If you're closing potassium channels, why would that depolarize? It's because potassium flows out, not in, so you're preventing the outflow of potassium. So we're not gonna worry about that. But anyway, you get neurotransmitter release anyway. So basically, I just want you to know you have activation of G proteins and second messenger systems that result in neurotransmitter release in that scenario. And then finally, bitter compounds like quinine. Quinine is a bitter substance. And bitter compounds, when they bind to their receptor, this also activates G proteins, which then activates second messengers, which results in neurotransmitter release. So you're still getting receptor potentials, you're just getting them in a different way by G proteins. And G proteins either opening or closing gates, depending on the situation. So again, neurotransmitter released, and there goes the sensory neurons, sending action potentials down the neuron. So if you go back to this picture, what we were just seeing was happening, here. This was being stimulated by some type of tastant, and that stimulates, receptor potentials, which stimulates neurotransmitter release. And then in this neuron, when it is stimulated to the threshold, it'll generate action potentials that get propagated to the central nervous system, which we're going to look at real quick next. Any questions about that? So, those second order sensory neurons are found in 3 cranial nerves. Those first order sensory neurons are found in the facial nerve, which is cranial nerve 7, the glossopharyngeal nerve, which is number 9, and the vagus, which is 10. And they are picking up taste sensations from different places. Facial and glossopharyngeal from the tongue, vagus from the throat primarily, like the epiglottis, for example. But just know that they're conducting taste information to the thalamus. So taste information is traveling through those nerves. You should know the nerves, but I'm not going to worry about where specifically they're coming from. But they all conduct taste sensations, sensation input to the thalamus, and from there it goes to the primary gustatory area. Now notice that in this sensation, there's no crossing over, just like with olfactory sensation. There isn't crossing over. In fact, I won't say all of them, but most of the special senses do not decussate. There is the exception of vision, but you don't have that decussation like you have with the somatic. Somatic sensations cross over in the brainstem or in the spinal cord. But notice that it's all staying on the same side of the body. It's what we call ipsilateral, all on the same side, not crossing over. Anyway, it goes to the thalamus. Remember, that's a relay station for every sense except smell. And then from there, there's a communication with a second order interneuron that sends that sensation to primary gustatory area in the lateral side of the cerebrum in the temporal lobe. And there's a picture of that area right here around this 43, about there at the base of the postcentral gyrus. So it's very close to, fairly close to where the olfactory area is, a little bit superior to that. But I guess it's actually in the parietal lobe, not the temporal lobe, but it's on the side there of the head, or the brain, I should say. Any questions about that? So primary gustatory area is what allows you to process those senses and recognize those tastes. But remember that taste is also a combination of smell and what you taste together. Plus, scientists are discovering new receptors. There are other receptors that we're not talking about. So there have been some new discoveries in terms of types of receptors, but those are the ones that are best understood or best known. Any questions about taste before we move on to vision, which is a lot more complex? Okay, any questions about taste? Okay, so taste and odor, odorants and tastants are chemicals. So those are receptors that we're talking about when you look at taste receptors and smell receptors. In the case of vision, we're looking at photoreceptors. Photoreceptors, which are the rods and cones that we'll be looking at, detect visible light. What they're detecting, when we talk about visible light, it is part of what we call the electromagnetic spectrum, the spectrum of radiation that travels in waves. That's what defines electromagnetic radiation. It consists of photons, which are little packets of energy, and these packets of energy travel in waves. They have different wavelengths. Some have very short wavelengths, some have very long wavelengths. So this is just showing you the different types of radiation and what we mean by waves. You know, like waves in the ocean, except these are photons or packets of light, or packets of energy. These packets of energy contain energy, and that energy can stimulate photoreceptors. Wavelength refers to, if you measure it from peak to peak or trough to trough, and that wavelength can vary from kilometers to meters all the way down to nanometers, which are very small. There's 1 billion nanometers in a whole meter, so we're talking, very small wavelengths up to very long wavelengths. So electromagnetic radiation is, for example, what the sun radiates. The sun radiates electromagnetic radiation, but so do some other things, including some instruments or machines that we build. We make X-ray machines and gamma rays and stuff that we generate. So it's these are still photons of energy that are being generated. Now, the wavelengths of the visible light spectrum range from 400 to 700 nanometers. Again, we're talking billions of a meter, fairly short, but not as short as gamma rays or X-rays. The color of light depends on its wavelength. So here is another view of electromagnetic spectrum. To give you, here's some comparisons of how large those waves are. Radio waves, which are very low, can actually be in kilometers as well as meters. Could be as wide as a building or even longer than that. And then you get into radio waves and microwaves, infrared, which we measure as heat. And then on the other range, gamma rays, X-rays, which are very high energy, short waves. The shorter the wave, or the higher frequency waves, which are shorter wavelengths, are also higher energy. So this is way more energy. Radio waves are very low energy. Gamma rays, X-rays, which are damaging types of radiation. Some radioactive material releases gamma rays, so it's very high energy, very damaging to tissues. Ultraviolet, which is some of it penetrates our atmosphere and causes tanning, but it also is high energy enough to cause DNA damage that leads to cancer, like skin cancer. So it also damages your eyes if too much of it gets into your eyes. Wedged in between ultraviolet and infrared is visible light, which is around the size of cells, I guess. Maybe a little bit smaller, I would think. But anyway, notice how much light gets through or how much of the spectrum gets through the atmosphere. Some radio waves, microwaves, some infrared actually gets through in the gray zone here. And what they don't show is that some ultraviolet does penetrate. Most of it is blocked out by the ozone layer, but there is some penetration where we wouldn't get suntanned. So there is some UV light getting through. But visible light gets through really well, which is why we've evolved to be able to see visible light. Some insects and other animals like owls can see ultraviolet light. We can't see that, but we can see the visible light spectrum. And just a real quick review of this from grade school. If you pass white light, which contains all the waves or all the wavelengths of the visible light spectrum, white light is all the wavelengths. If you pass it through a prism, the prism splits the light, bends the light and splits it up into the colors of the rainbow that you can see make up the visible light spectrum. And so when you see, the rainbow in the sky, that's from that prism effect of air, of water or moisture in the air, and the sunlight hitting it just right, creating a prism effect. And you may have learned in grade school the order of the colors: red, orange, yellow, G for green, blue, and indigo, and violet, which indigo is really just blue or violet, I guess. But anyway, here is those wavelengths again on their side, long waves for radio waves and so forth, gamma rays, very high energy, and wedged in between the high energy ultraviolet radiation, which can cause cancer, and infrared, which is heat, that we feel as heat, is the visible light spectrum. It's got enough energy to do things like power photosynthesis for plants. It also has enough energy to activate our photoreceptors. So it's not too much energy, it's not too little, it's kind of in the right Goldilocks conditions in terms of activating photoreceptors. This range is 400 to 700, and you might be surprised to learn that violet and blue, which we consider to be cool colors, are actually more high energy. They are on the ultraviolet end. So they are more high energy light, which is why you often hear about blue light coming from your phone and computer screen, damaging your eyes or straining your eyes or causing you to lose sleep at night. And that you put these blue filters on your phone at night so it's not hurting your eyes or overstimulating your eyes at night, which could interfere with sleep. It's because blue light is on the short wavelength side, so it's more energetic. Red, on the other hand, is on the infrared side, so it actually has less energy, even though we consider red to be a warm color. If you were looking at a blue flame versus a red flame, the blue flame of a fire would be hotter than a red flame is. So this is actually more high energy than this is. Green falls right in the middle. So the longer the wavelength, the less energy. The shorter the wavelength, the more energy. And you have what are called cones, photoreceptors in your eye, that are activated by these different wavelengths, creating the different colors that you see. Now we're going to look at the external structures and then the actual structures of the eye, and then look at how the eye works. Accessory structures just refers to outer structures that help the eyes do their job. I'm not going to have you learn all the parts that are in here, but a few things you should know include palpebrae, which refers to the eyelids. The upper eyelids and lower lids are called the palpebrae. And they, of course, protect your eyes from various things getting into them, and also spread tears on your eyes. You have the eyelashes and eyebrows, which also protect your eyes, keep sweat out of your eyes, and help screen some of that sunlight, and also prevent other objects from getting into your eyes. So these have protective functions. There's also the conjunctiva. This is a protective membrane inside the eyelid. It lines the eyelid and covers the white of your eye. What's called the sclera is the white part of your eye, and it actually has this membrane over it, the conjunctiva. So it's not just the pink, when you pull your eyelid back and you see that pink tissue, well, that's the conjunctiva, but it's on a layer of blood supply, so it has that pink color to it. But the conjunctiva is clear on the sclera surface, and so you don't usually see it unless you get, some blood vessel breakage, and then you might see red in that conjunctiva. Sometimes you get, or if you get pink eye and you have, pink there, that is the conjunctiva. It's not the white of the eye usually that's got the blood vessels that are broken. It's usually the conjunctiva, or if there's inflammation. And so the conjunctiva, and I'll show you some other pictures of that, it is a mucous membrane. It consists of non-keratinized stratified squamous epithelium and goblet cells. The goblet cells secrete a mucus layer on the eyes. And this covers the areolar connective tissue below, which together makes up the mucous membrane, epithelium on top of areolar connective tissue. You may have heard of pink eye or conjunctivitis, and that is inflammation of the conjunctiva, usually due to a bacterial infection, and it can be quite contagious. So here's a picture pulling the eye back. The palpebral conjunctiva. The palpebra is the eyelid. The palpebral conjunctiva is that what looks like pink tissue under the eyelid, and also if you pull that eyelid back, you'd see it. And the bulbar conjunctiva is transparent, and it's covering the white. I'm not concerned about the names of the conjunctiva, but you should know where the conjunctiva is located. And in this side view, this section view, here's the eyelids or palpebrae. The pink is the palpebral conjunctiva. It lines the inside of the eyelid, and then the blue is the bulbar conjunctiva that covers the sclera of the eye. And this protects the eye. It's also part of the tear production apparatus. We think of tears as just being the salty, watery tears that we produce, but there's actually 3 tear layers in what they call the tear film. And this is important in getting rid of, helping wash away irritants in the eye, keeping the eyes moist, which helps with good vision. And it keeps the surface smooth so that you don't get scarring or anything in this conjunctiva. If you don't have enough tears, that can cause scratching of the conjunctiva and irritation and all that. So the tear film actually consists of 3 layers. There's an outer oily layer, a middle watery layer, which is produced by your tear glands, and then the mucus layer, which is produced by the conjunctiva. So I'm going to start from the inside out because we were looking at the conjunctiva, but the inner layer of tears is the mucus layer, and that's produced by the goblet cells in that conjunctiva and that mucous membrane. And that not only keeps the eye surface moist, it also helps the tears stick to the eye. The mucus actually attracts the water of the tears and keeps it on the eye instead of allowing it to run off. If you don't have enough of a mucus layer, your tears would kind of run out of your eyes. You'd have runny eyes, and that can happen. In some cases, people have tear problems, tear production problems, and if there's any problem in any of these layers, it can cause other side effects. So if your mucus layer if you don't have enough of a mucus layer, your eyes would get watery all the time. So there's the mucus layer, which allows the watery layer to stick to the eye. The watery layer is what's produced by the lacrimal glands, which we're going to look at shortly. And this is a salt water solution that helps wash away irritants. And then you have, on top of that, you have the oily layer. This prevents the tears from drying too quickly. Some people have problems with the meibomian glands that produce this oily layer. And we'll see where those meibomian glands are, but they're basically located right along the rim of your eyelids, right behind the right behind the eyelashes. And these meibomian glands produce this oily layer that prevents the watery layer from drying up too quickly. And sometimes the ducts of the meibomian glands get blocked, and that prevents you from having that oily layer, which leads to dry eye. So one cause of dry eye can be meibomian gland problems. So here's a picture again of side view, sectional view of the eye. Here's the eyelids right here, the palpebrae, and here in green are the meibomian glands that release their secretion just behind the eyelashes, And that oily layer helps hold on to your tears and prevents dry eye. Now, lacrimal glands are what we consider to be the tear glands. I'll show you where they're located. They secrete the watery layer of the tears, and they contain water, salts, and lysozyme, which is an antibacterial enzyme, which helps prevent infection. And of course, this keeps the eyes clean. It helps wash away irritants and lubricates the eye. You only produce about 1 milliliter a day of tears, which is why you need that oily layer to hold on to that 1 milliliter. And when you blink your eyes, the tears move medially. They're actually, the tear glands, as we'll see, are up here in the lateral superior part of your eye orbit, just underneath the ridge of bone here, kind of between the bone and your eye. You've got these tear glands, and I'll show you those in a second. And they drain into, the tears drain from the eye to the nasolacrimal duct. So here in green is the location of the tear glands, they're kind of above the eye on the lateral side, wedged in between the bone and your eye, just above your eyelid, and your upper eyelid, and they're releasing tears laterally, and then the tears wash medially and then drain down these ducts to the nasolacrimal duct, which carries tears to the inside of your nose, which is why if you've got irritants or allergies or you're crying, and there's an increase in tear production, it makes your nose run, because those tears do run down your nose, and that helps wash away irritants. That's also why you get that kind of eye crud at the corner of your eyes sometimes after you've slept, because the mucus collects there and other stuff, particles and stuff collect there and get caught kind of in the corner of your eye where those ducts are. So that creates that. Your tear glands are produced or stimulated by your parasympathetic division of the autonomic nervous system. And so the parasympathetic division increases tear production, especially if there's any irritation or allergies or anything like that, and also if you're crying. Now some other accessory structures of the eye. Of course, this is a lateral or a sectional view, looking at the lateral view of the eye. And we're going to be looking at the eyeball in more detail, but the white of the eye that I was talking about, which is called the sclera. The sclera extends from the iris. So here's the front of the eye. This is called the cornea, this clear covering the front of your eye is called the cornea. It's what covers the iris and pupil. So the blue here is the iris. That's the colored part of your eye. You have green or blue or brown eyes. And then the pupil is the opening, which can increase or decrease in size to control how much light goes into your eye. Here's the lens, which is a transparent lens that focuses light as it comes in and focuses it to the back of your eye. And it is controlled by little fibers that are connected to smooth muscle that either make the lens rounder or less elongated. We're going to talk more about that later. And then around the, when you get past the cornea, the posterior part of the eye, after the iris, you've got the white of the eye, which you can see the white of your eye, but it extends all the way to the back of your eye. The white sclera is what it's called. The sclera of the eye extends all the way back to the optic nerve. So there's the optic nerve, and that white layer then extends out as a connective tissue covering around the optic nerve that transmits vision or photo information to your brain. There's some other structures you can see here. Of course, the eyelids we talked about, or palpebrae, the meibomian glands, and so forth. There's a lot of fat in the eye orbit, which protects the eye, especially in the very back of the eye. A lot of fat, orbital fat, that protects the optic nerve, and also keeps the eye, from moving around too much there inside the eye orbit. And you can see some muscles, for example, up at the top there, the levator palpebrae superioris. This tells you it involves the eyelid. Levator means to elevate, and superioris means it's above the eye. So it tells you a lot about the muscle. This muscle is the muscle that is attached to the upper palpebrae or superior palpebrae or eyelid. Palpebrae means eyelid, that helps. And it pulls on the eyelid and opens the eye. So the palpebrae or levator palpebrae superioris is the muscle that pulls your eyelid up. The other muscle that is involved in moving the eyelids is the orbicularis oculi. You may recall that's a circular muscle around the eye that is involved in closing the eyes and also squinting and blinking and things like that. And we'll see another picture of that one in a minute. Some other muscles that are involved with the eye are the extrinsic eye muscles. Extrinsic meaning outside the eye. They are muscles like the superior rectus and the inferior rectus and some other muscles that are involved in moving the eyeball and moving your eyes around. So those are extrinsic eyeball muscles. So we're going to look at some pictures of that real quick. Here's the orbicularis oculi on this side. This is the superficial side of the picture. And these muscles, like I said, you can activate different parts of it. You can blink the eyes, you can close them, in other words, or you can also squint with this muscle. And then over here, when you remove that, you can see this is a little bit of that muscle, the levator palpebrae superioris, that pulls on the eyelid and opens the eye. You can see it's located, it's a superior muscle. It's actually, if you go back here, it lies over the superior rectus muscle. So it sits on top of that muscle. And then off to the side here, just above that muscle, in the lateral wall here, is the lacrimal gland. So the lacrimal gland is yellow here. You can see where it's located. And here are those extrinsic eye muscles. You may recall the superior, well, let me first say, you may recall that all but one originate at the back of the eye. They all originate on what's called the common tendinous ring, and they all insert on the sclera, or the white of the eye. They pull the eye in different directions. I'm not going to worry about the different muscles, but just know them as the extrinsic eyeball muscles. All but one, this one here actually originates in the corner of the eye down here and attaches like that. That's the inferior oblique. But these muscles, there's 6 of them, and they move the eye in different ways, you know, allowing you to move your eyes around in your head. So those are under your somatic control. Now we'll see that there's also muscles inside your eye. Those are intrinsic muscles, and those are actually smooth muscle, not skeletal. You know, these are, of course, skeletal muscle. And we'll see those in a minute, but let's look at the 3 layers in the wall of the eyeball. There's the outer or superficial fibrous tunic. The vascular tunic lies under that. It's also called the uvea. And then the inner layer is the retina, the back of the eye. And we're just going to work our way through this. The fibrous tunic is called that because part of it is very fibrous. The sclera, the white of the eye I was just telling you about, makes up that outer fibrous tunic, at least when you get behind the cornea. And it has, it's dense connective tissue, dense irregular connective tissue with a lot of collagen, which is why it's white. You may recall that collagen is a white protein fiber, and it gives tissues that contain a lot of collagen a white appearance. That's why the whites of your eyes are white. And the sclera is very strong. It's thick and strong, and it helps maintain the round shape of your eye and prevents the eye from collapsing. So it's a very strong structure, very rigid, and it's also where the extrinsic eyeball muscles attach that move your eye. And then you have the transparent layer in the front, and that's called the cornea. And this covers the iris and lens. It is a curved structure, so it helps in focusing light. It is not the primary agent of focus, the lens is, but it is transparent. It does bend the light a little bit when light passes through it. And so let's look at these structures again. The white of the eye that I've talked about extends all the way to the back of the eye, you know, all the way to the optic nerve. That's the sclera. And of course, it's covered in the conjunctiva that you can't see. And then here's the cornea is here. You can't see it because it's transparent, but it covers the colored iris and the pupil where the light goes through. So in this picture, you have the layers of the eye. The eye has been cut open. You can see the 3 layers of the eye. We're going to talk about the other layers in a minute, but here's the sclera, that thick collagen-filled connective tissue, and it extends all the way to the iris, where then it's replaced by the cornea. But they are all, you know, they're connected. The cornea, you can see, is rounded in the front, and behind the cornea is the iris and the lens of the eye. This is where the light is coming through, you know, into the eye like that. So the iris, there's the pupil in front of the lens. Okay, the next layer is the vascular tunic. It's also called the uvea. It's the middle layer between the outer fibrous layer and the retina. And there are different parts of it. There's the choroid, the ciliary body, and the iris. The iris is part of this layer. And we're going to look at the different parts first. The choroid, which is in the posterior wall of the eye. It lines most of the inside the sclera. So basically where the sclera is, just under the sclera part, you have the choroid. So the choroid would be under the sclera, not the cornea. It is very vascular. In fact, this is why it's called the vascular tunic, because there's a lot of blood vessels in this part of the eye. It does provide the blood supply to the retina, and it contains melanocytes. You may think, well, melanocytes are in your skin, and you know that's what causes your skin to have its color. Melanocytes also give your hair and eyes, the iris of your eyes, their color, but you also have melanocytes deeper in your body, including in this choroid layer. And this is what makes it very dark brown, and one reason why the inside of your eyes are so dark, because of melanin. This melanin helps absorb stray light rays, so you don't have light bouncing around inside your eyes, and you know, overstimulating your photoreceptors and creating too much glare and pain, you know, if there's too much light and things like that. So it helps in absorbing some of the stray light rays inside the eyeball. So it has a protective function. And the choroid layer is shown in this picture. Even though it's really dark, they're showing it pink for some reason. Maybe so it just wouldn't be too much, but the choroid layer extends from about here. It's where the dots are, the pink dotted layer here. The dots are actually red and blue little blood vessels. That's the blood supply. But all of that, you can see most of the sclera is lined in the choroid layer, which again has blood vessels, and it has the blood supply of the retina, and it also has, which you can see the blood supply here as well. That would be in the choroid layer. And it's dark, so it absorbs those stray light rays. So working from the outside in. But there's some other structures as well in the uvea, or in that vascular layer, that vascular tunic. If you keep going forward into this pink and kind of reddish brown area, you get to what's called the ciliary bodies on each side, and they are located on either side of the lens. So the ciliary body, which is more anterior, it contains melanocytes as well that produce melanin, and it says it's dark brown. It's almost black. The ciliary body is very, very dark, and it's also helping to absorb some of those stray light rays. The ciliary body contains the ciliary processes, which we'll look at in a second. These produce aqueous humor fluid, and the ciliary muscle, which is a circular band of smooth muscle around the lens that contracts and pulls on zonular fibers, which change the shape of the lens and focuses the lens. So you can see those structures a little bit here. Here's the ciliary body on either side of the lens. And it's got these little processes that stick out. Those are called the ciliary processes. There's also smooth muscle, the ciliary muscle, which runs in a circular ring around the eye. If you went around the eye, around the lens, you would have that ciliary muscle. And the ciliary body is attached to these thin fibers, which are protein fibers that attach to the lens. When the muscles contract, that pulls on those fibers and makes the lens thinner. When they relax, the fibers relax, and that makes the lens thicker. We'll talk more about that in a minute, but it helps in focusing the lens. So that's how you focus the lens. And let's look at it more close up. So this is a magnified image of what we just saw. There's the lens, there's the iris, which we'll get to in a minute, and behind the iris, just behind your iris, is the ciliary body. The ciliary body consists of ciliary processes. These things have capillaries in them, and the capillaries filter blood and make aqueous humor, which is the fluid that circulates through these chambers here in the front of your eye. The fluid here, and in this anterior chamber here behind the cornea, you have aqueous humor. This is a watery fluid that circulates around those areas. And then you have here the ciliary muscle. Here's part of it here, and here's some of it here. This is cross-sectional, this is longitudinal. So the ciliary muscle, when it contracts, it pulls on these zonular fibers, and that can pull on the lens and make it elongate. When the ciliary muscle relaxes, the lens becomes more round. So you're either contracting or relaxing and controlling the lens shape, which then helps with focusing, as we'll talk about shortly. Let's see, so that's the ciliary body. Any questions about that? That was back here. Let's see see if I'm missing anything. I think I've covered all that. So let's look at the iris. This is the part of your eye that you can see. This stuff is behind the iris. You can't see it. But this part you can see. And the color of your eyes depends on melanocyte activity. You inherit from your parents the genes that control how much melanin you produce and the type of melanin you produce. And so the more melanin you have, the darker your eyes are going to be. So if you produce little or no melanin, you might have blue eyes. If you've got a little ring of gold, that's a little bit of melanin. If you have more, golden eyes, that's even more melanin. If you have very dark eyes or brown or almost black eyes, even more melanin. If you have green eyes, it's really a combination. It's that melanin on a blue background because your iris has a blue layer in it. And so if you have that golden melanin on the blue background, it looks green, gives you a green-eyed appearance. There's not a green pigment, it's just the combination of the melanin, which is kind of golden color, with the blue background gives it a green appearance. So all the color combinations that we have in our eyes depends on which genes you inherit. There's more than one gene involved in melanin production, so that's why we have such a wide variety of eye color. The purpose of that melanin is to protect the retina, to protect the eyes from UV radiation. It helps block some of that light from getting into your eyes and damaging the retina. And so, the darker the pigment, the more protection there is. In people who have certain types of albinism, who are albino with pink eyes, they have no melanin and no pigment at all, not even the blue pigment in their eyes. And what that pink you see is actually pink from the retina. The retina is very vascular. So that pink color is that reflected light from that red of the retina, which means the retina is unprotected. So they have to wear sunglasses a lot to protect their eyes. Now there's also, in the iris itself, you not only have pigment, you can't tell this, but you actually have smooth muscle fibers, radially oriented and circular smooth muscle that can either constrict and close not completely close it, but constrict the pupil or dilate it. So let's look at that real quick. So here again, the sclera, or the cornea is the clear covering, and the iris is this colored part, the pupil is that. And in your pupil, or in your iris, I should say, here's the pupil, and here's the iris smooth muscle. You have radial, meaning the smooth muscle radiates outward this way, and you have circular. The smooth muscle runs in a circle around your iris. Here is regular, relaxed pupil that isn't either constricted or dilated. This is just under normal light conditions. If you are exposed to more light, or if you're reading or doing close-up work, your parasympathetic division, and somewhere it talks about that, your parasympathetic division controls the circular muscle, the circular smooth muscle. And that when it is stimulated by the parasympathetic division, it contracts, and as it contracts, that smooth muscle contracting actually constricts the opening. So you have less light, and that way you protect your eyes if it's bright outside. But you also, if you look at someone when they're reading, they're going to have more constricted pupils. That's a parasympathetic activity controlled by oculomotor nerve. This nerve controls extrinsic eyeball muscles, but it also has a parasympathetic control. This is also called cranial nerve 3. And then you have, on the other side, radial muscles are controlled by your sympathetic nervous system. And when the radial muscles contract, they pull on the circular muscles, they pull it out in all directions, and that opens up your pupil and allows for more light in. So under dark light conditions, this happens, but also when you're under sympathetic stimulation because of an emergency, or you're exercising, or you're doing something else, your eyes will dilate, and this allows you to see far away. It allows you to see further away. Not so much close-up vision, but further away. And that, in an emergency, you might need to run and be able to, have far vision for running or escaping a predator or something like that. So it has that function. So these can constrict or dilate depending on light conditions, but also depending on what you're doing at the moment, whether you're doing something up close or whether you're you're running, you know, for exercise or something like that. Any questions about that? So 2 layers of smooth muscle in your iris. So that would be one of those intrinsic muscles of the eye, inside the eye, and they're smooth muscle. Now we're going to look at the retina. This is taken with an ophthalmoscope. I always have a hard time saying that word, but anyway, this is that scope that they use, you know, with the bright light that goes in, you know, that they look at your eye with. And when you look at the retina, you can see it's very red because the choroid layer has a lot of blood vessels in it. Really what you're seeing is the choroid blood vessels here. All these little blood vessels are in the choroid layer, but the retina lies over the choroid. And in this you can see there's what's called the optic disc. We're going to talk about that in a minute. That's where your optic nerve is, but also where the arteries or the arterioles and venules enter and exit the eye, or arteries and veins exit and enter the eye. We're also going to talk about this little region here called the macula. We'll get to that in a minute. But this is, you know, this is what they're looking at. Sometimes when they're shining that light in your eye, you can actually see those blood vessels. You might get an image of those blood vessels because they're lighting it up so much that it creates a shadow of that light. So here's where that optic nerve that I just showed you, that's this area right here is where the optic nerve, but also blood vessels enter at that layer. So you've got the optic nerve. It surrounds the retinal arteries and veins that supply blood to the choroid layer, which in turn supplies blood to the retina. See if there's anything else I want to point out here. We'll come back to that in a minute. So the photoreceptors, and this is just a drawing of that ophthalmoscope image. Photoreceptors are in the retina. That's where vision is sensed. And here you can see blood supply. You can see the optic disc where the blood vessels are entering the eye. And this is also where the optic nerve is. And by the way, this is also an area where there are no photoreceptors. The optic disc it does not have photoreceptors. So this is what's called the blind spot in your eye. You don't notice the blind spot unless you know, there's a thing in your book about how you can find the blind spot, but it's you have 2 eyes and you kind of compensate for that. You don't actually notice a blind spot unless you do a little test for it. But that there is a small area in the retina that doesn't pick up any light image. The macula lutea, that's what you're seeing right here, that little kind of orangey looking area. The macula lutea is a yellow area at the back of the eye. And it's yellow because it contains 2 pigments, lutein and zeaxanthin, which I mentioned in here somewhere in the notes. But the macula is an area that surrounds the fovea centralis. The fovea centralis is a little pinhole-sized area where basically you're focusing all your light. This is the very back center of the retina. The light that you're focusing is being focused on that little point. It's a pinhole-sized point. And that's where, you know, like when you're focusing on a face or on a book or whatever you're looking at, words on a page, that's where you're focusing everything. You might notice that everything else is less clear than what you're focusing on. You're focusing on one person's face, everything else is kind of peripheral to that. And that's what you're focusing on. And you want to keep that nice and healthy. The macula contains yellow pigments. They're called lutein and zeaxanthin, and they're in my notes somewhere further back. But anyway, those pigments you can get from eating yellow fruits and vegetables, and also green leafy vegetables like spinach and kale contain high amounts of these. And this accumulates in the macula, which gives it a yellow color. What it actually does is it apparently protects the fovea centralis and these photoreceptors from blue light, from that damaging blue light, and maybe also UV light, I'm not sure, but it protects from that damaging blue light, which shows you how important or how bad blue light is, that you have to have some region that protects you from it. But this is one reason why you want to have blue light, you know, screens, especially at nighttime when you're looking at your cell phone or a computer screen. Anyway, that area is also where, if you've ever heard of macular degeneration, that is degeneration of the macula. And one way of treating that, or even possibly preventing it, is these AREDS vitamins that you see marketed. They're actually been shown to help improve or prevent further macular degeneration. But macular degeneration results in loss of that central vision. You know, you just basically have no central vision, everything's peripheral. And it can be very, very difficult. So a lot of people have a genetic tendency toward that. It might run in your family. If it does, you may want to talk to your doctor about taking AREDS vitamins as a preventative, maybe not taking the full strength, but maybe half strength or something like that, if it does run in your family. But it is a fairly common cause of blindness in elderly people. Not something you want to have to deal with when you're older. Here is the location of the macula lutea. It is right center, right behind the lens, straight back. And that fovea centralis, it's kind of a cup-shaped area where the fovea centralis is. And it's just that little point right there in the middle of that. So it's kind of cupped inward where the macula is. And at the center of that is the fovea centralis. And you have a high density of photoreceptors in that area. So that's where your central vision is right there. And then just a little bit, I guess, lateral to that is the optic nerve. I think that's right. So, let's see here. So there there's the thing about lutein and zeaxanthin. So that's how you spell those words. But anyway, this is the left eye. I should say medial. Sorry about that. Not lateral, medial. The nasal side is where toward the nose is where your optic disc is. Further lateral is where the fovea centralis is. So this is, I guess, the right eye. But anyway, so that's more nasal and that's more lateral. Okay, what else? See if there's anything. Actually, fovea, did I have it? Fovea centralis, and I have fovea capitis. I need to fix that. Anyway, that I'll fix in the notes and put it back up. The lens is a connective tissue capsule that is transparent. It consists of what are called crystalline proteins. They're called that because they're clear proteins, like a crystal, and they're arranged in layers like an onion. So you have these layers of this connective tissue protein. And so when it's clear, of course, it's going to work best. But cataracts occur when you start getting oxidation of these proteins and you start getting cloudiness in those proteins. And that's what happens with cataracts. And it makes a cloudy vision. And sometimes people have to have that lens replaced with a new lens. But nowadays, technology is such that they can give you a lens that works better than your old lens. So it's not such a terrible thing if you have to have corrective lens surgery. But like I said, the lens is transparent also because it does not have blood vessels in it. No blood vessels in the lens. And the lens divides the eyeball into two cavities, the anterior and posterior. So the lens, everything next to and in front of the lens is the anterior cavity. The posterior cavity is here. The anterior cavity is also further subdivided into an anterior and posterior chamber by the iris. So here's the anterior chamber of the anterior cavity and posterior chamber. Everything behind that is the posterior cavity. We're going to look at this in more detail. Let me find a picture. This is just talking about pressure and all that. But again, remember I told you the ciliary process of the ciliary body produces the aqueous humor. The aqueous humor is released into this posterior chamber of the anterior cavity, and then it continues to circulate into the posterior chamber. And about 90 minutes later, here, follow these arrows here, the aqueous humor gets reabsorbed into a venous, small venous sinus or vein called the scleral venous sinus. And this is a tiny opening at the very outer edge of the iris, the scleral venous sinus. And that sinus is also called the canal of Schlemm. It's another name for it. Now, it's important that you're producing and reabsorbing that aqueous humor it's called the aqueous humor, but it's a fluid. The aqueous humor is, you want to produce just the right amount. You want it reabsorbed as well, because if not, it can increase your intraocular pressure. Your intraocular pressure should be around 16 millimeters. There's actually a range that's normal, 16 millimeters of Hg, which is mercury. You know, in the doctor's office, they test your pressure, they touch that thing to your eye after they numb your eye, and that's what they're measuring. They want to make sure it's not too high or too low. Too high pressure in the aqueous humor in that anterior cavity can put pressure on the back of the eye and cause optic nerve problems that lead to eye loss, or not eye loss, but vision loss. That's called glaucoma. So too high a pressure can cause glaucoma, but on the other end of it, too little pressure can cause the retina to detach, because the pressure inside the eye helps hold the retina against the back of the eye. The retina can actually peel off the back of the eye, can peel off the choroid, and when that happens, you've got a detached retina, which is no good. So you have to have surgery to fix that. So again, this is where you find the aqueous humor, that fluid that creates that internal pressure. But there's also a posterior chamber called the vitreous chamber. It's called that because it contains a gelatinous vitreous humor, and we'll look at that as well. It's also called the vitreous body, vitreous humor or vitreous body. This is more jelly-like. It's transparent. It also helps to hold the retina against the choroid. So it's kind of, you know, along with that pressure from the aqueous humor, it's putting pressure on the retina and keeping the retina up against the choroid layer so it doesn't detach. So that's important. The vitreous body or vitreous humor is produced during embryonic development. So it's not constantly being produced and reabsorbed. You make it as an embryo and you have it for life. The problem with that is, because it ages with you, you start getting problems like sometimes the vitreous body will contract and liquefy, and that causes retinal wrinkling or retinal detachment as people age. So that's not uncommon for that to happen. Also, the vitreous body contains, it's gelatinous because it contains hyaluronic acid, which gives it kind of a slimy, gelatinous character to it. And it also contains collagen. And sometimes you see those little floaters, those little shadows in your vision. These are clumps of collagen that cast shadows on your retina. So those floaters are actually collagen. That's usually, you know, usually it's collagen anyway. There's also white blood cells that are there to consume foreign things like that collagen and to consume bacteria. Okay, so this is where the vitreous chamber or posterior cavity is. It's called the vitreous chamber. It contains that vitreous body, which holds the retina here in yellow against the choroid. This is the retina in yellow. It's not really yellow, but that's the picture. You know, the picture they got is showing it as yellow. And by the way, the retina would extend, you know, all the way to the iris. All right, so let's move on. So this table, I'm not going to go through it, just summarizes the different parts of the eye. You might want to find it helpful to look at that table on your own when you're studying. Now what we're going to do is look at the image formation, how you form an image, how you focus the lens. And it requires three things. Bending of light, which is called refraction, and this is done by the lens as well as the cornea, mostly the lens, but the cornea helps too. Accommodation, which is the change in shape of the lens during focusing. The cornea doesn't change shape, but the lens does. And then constriction or narrowing of the pupil, which, as I mentioned, not only does your pupil constrict and dilate based on light coming into your eye, but based on what you're doing. So close-up work, you're going to constrict the eye. If you're driving and looking in far distance, or you're running, or you're doing something else and you're looking away, 20 feet away or more, then your eye is going to dilate. So we're not going to spend too much time on this part, but we're going to spend a little bit more time on the refraction and accommodation part of this. Now, light bends when it passes through things like water. So here's refraction of light. And, you know, here it is in the air, and then if it hits water, it refracts. But it also does that when it passes through the cornea and lens. So the cornea and lens are like water. They bend or refract the light. So the cornea, the outer clear covering, it's round, and as a result, it's going to bend the light a little bit, and then the light gets bent even more by the lens. Now, this is showing, and that's called refraction, that bending of the light by those two structures. Both of these are convex. What that means is they're rounded outward. Concave, they'd be rounded inward, but they're rounded outward. And that rounded outward convex shape allows them to bend the light. Now notice that if you're looking at an object, when the light is bent by the cornea and lens, it's bent to the degree that it actually turns the image upside down. In other words, those light rays cross before they hit the retina, and so everything is upside down and backwards. In other words, left, right, reversal. In other words, if you're looking at, let's say you were looking at a letter E on your retina, the image on your retina would be like this. It would be reversed. It would be upside down and backwards. Now, we don't see things upside down and backwards though, do we? We see it right side up. And that's because your brain turns it all over, turns it right side up again. And this happens during infancy. Your brain learns to reinvert, because if a baby's reaching for something and it's actually over here, it's gonna learn, its brain's gonna have to learn to reverse everything. So your brain magically somehow manages to figure out how to reverse those images. So what we see is the way it is, yeah.
Did your brain figure that out before the baby's actually born and starts seeing, or are babies born seeing upside down?
I think it is that they're born seeing things upside down and backwards.
Uh-huh. That is neat.
I'll double check, but I'm pretty sure, that this is something they learn as infants. But they're so young, we don't know. You can't. But that may, explain why they, are not very coordinated when they're reaching for things. But that's also muscle control and all that. But I'm pretty sure, it's when they're very young infants when they learn that. It is pretty amazing. Okay, so let's look at what happens in far vision. And this would be over 20 feet or more away is what we're talking about. Something that's 20 feet or more away. So you're looking at a distant tree, and like I said, the image gets inverted, upside down and backwards. But also look at what's happening. Light coming from images far away, though that light travels almost parallel to the ground. So it doesn't take much to focus it or to refract that image. So when you're seeing something far away, the cornea and lens are able to just bend the light with very little accommodation of the lens, bend the light so that it's focused. And this is supposed to be where the fovea centralis is, right here. That's where it's focusing the light. If you're focusing on a tree or a car, or you're driving along, you see a car coming towards you in the other lane, that's what you're focused on. That's what you're looking at that moment, and it doesn't take much to bend the light to get it focused on your fovea centralis. And so notice that the lens is elongated. The lens is elongated at that point. And again, the fovea centralis is where the vision is sharpest. That's your center of vision that you're focused on. Yeah.
So people who can't see far away, what is like the main?
Oh, we're gonna look at that in just a sec. That's next up after this. Oh, maybe it won't be today, but we'll be coming to that. So, again, this is just showing you the fovea centralis. That's where you're focusing that image. Now, and we still got a couple minutes, so hold your horses. Now, if it's less than 20 feet away, notice what's happening because it's closer up, like if you're looking at a tack, there's a thumbtack. The light rays are divergent, not parallel, and then the cornea refracts that just a little bit, but the lens is going to have to bend to focus that on that fovea centralis. So your lens has to bend inward. Here it's more relaxed and elongated. Here it's bent more inward and rounder. It has to get rounder in order to focus that light on the fovea centralis. And that's if it works right. So changing the lens, bending the lens to accommodate that's called accommodation, when you're bending the lens and making it thicker to focus on something up close. You're changing the shape. And that depends on the ciliary muscle. It contracts and relaxes here in that ciliary body, pulling or loosening those zonular fibers. and either letting the lens be more elongated for far vision or rounder for close vision. Notice it says that when the zonular fibers tighten, when the muscle contracts, it's going to tighten those fibers. The lens elongates because it's pulling on it. It's pulling it elongated. So when you're looking at far vision, you're pulling those zonular fibers. You're contracting the muscle, pulling the fibers, elongating the lens. When you're focusing up close, you are loosening those fibers, allowing that lens to change shape to a more round shape. So here, you're tightening the fibers by contracting the muscle. Here, you're loosening the fibers, allowing it to become rounder. And that's where vision problems often arise, is getting that image focused, just right on the fovea centralis. So accommodation, increase in curvature to focus on near vision and near point of vision. The minimum distance you can focus on something up close, which for young adults is usually about 4 inches. But as you age, that minimum distance gets further and further away. That's why people have to hold the books away. We'll talk about why that is next time, why your near point of vision changes as you